Welcome everyone for this PYQ discussion for test 3. Okay, so today we'll see the test 3, some of the questions. So these are from our own notes. So the first question which we'll discuss is the role of the plant hormones in agriculture and horticulture. Okay, so this is a very important question repeated every few years. Every 2 to 3 years once you will find this question. Okay, and indirectly or directly it is asked. Some of the important things which you should remember is like the role of auxin. It has an use, okay, in the treatment of cuttings, and also another very important is in the tissue culture. It is very useful. Okay, in tissue culture techniques, we use auxin. Okay, and also for the prevention of pre-harvest drop of fruits, we are using auxin, and also it increases the parthenocarpy. And the other advantage is 2,4-D, which is at higher concentration. It will act like a weedicide. Okay. So the next is role of gibberellins. So gibberellins are used to break dormancy. Okay, and also they induce uniform crop emergence, especially in case of seed potatoes. So I have told you many times. So when you read with example. One is it breaks dormancy and induce uniform crop emergence. Example in case of seed potatoes. Also, it is used to enhance the growth of vegetative parts of several floriculture species and increase the flower size. Here, there is nothing much to explain, just need to remember. Induces flowering in long day plants and promotes male flower production. So, you should remember that gibberellin is useful in male flower production. Okay, in general. Next, if you see about role of ethylene, ethylene is a hormone which is also known for the fruit ripening and it causes abscission like ABA. This is also involved in abscission. Okay, abscisic acid. And also this induces bulbing in onion. And also ethylene is playing a key role in tillering in rye and peas. And as you saw here it induces male flower production, but ethylene is involved in female flower production. Example is cucumber. Okay, and induces uniform flowering and ripening in pineapple. This also is important. It is useful in pineapple. Okay. Just you need to remember. And our students, we write only this much. We will write ABA, then other we will write uh, cytokinin. Okay, like in cytokinin, we can say its use in the tissue culture technique is also very prominent. And we should not restrict ourselves to these. We should go for next level, like in case of psychosol. Okay, it is used to increase the number of roots and tillers and prevent lodging and increase yield in case of wheat, rye, and oat. And also, psychosol is used to increase bloating and flowering and increase yield in cotton. Okay, and also remember this like florigen, it is useful for in flowering in plants, and this is also very important. Glasophile. Okay, so it is used in ripening of sugarcane. Next, this malic hydrazide. This is very commonly used and very important because to extend the storage life of bulbs and roots, we spray malic hydrazide. Okay, so these all things you can remember how these things are useful in plant growth and promoting different characteristics. If you remember this and if you are able to remember like 50-60% of this, still it will be very useful. Next, let us see about vernalization. You know about what is vernalization, what are the features. Those things will not be a concern. You can see from the best copies also which I have provided. But the practical benefits we are not properly writing. So the first thing is it is used to shortening the vegetative period of crop. What happens? What are the advantages? You shorten the vegetative period of crop. Okay, so you can have more crops per season, more crop per year. Okay, so indirectly that will also increase our production and also our profit. So should interconnect like this. Okay, the next is crops can be used in regions where they do not reproduce naturally, because naturally, every plant has its own uh, temperature width, okay, within which this will survive. So if you are giving vernalization treatment, then it can be also be produced in regions where they do not occur naturally, and it can escape frost, drought, and flood. Okay, because as it will shorten the period, okay, so there will be manlo in some season, next it will come frost in some areas, so it will escape frost and it will complete its life cycle in a very short period. And also it can accelerate the plant breeding, okay, and it increases cold resistance. Also there are reports which says that it has like fungal disease resistance also increases. And this you should remember, like in Russia, okay, the harmful effect of cold can be improving the thus improving the crop production. Okay, in case of Russia, this vernalization technique is highly useful as it is a cold region. Okay, like this, these are the major points which you need to remember and write. Next question, which usually is asked about breaking of dormancy. Okay, so if you're writing points just like that, some points, it will not look good. So better is like 1, 2, 3, 4, 5 classifications. One is scarification, wherein we are using physical or chemical treatment which will soften the seed coat. Okay, this will soften the seed coat which is there. This is known as scarification, and it is used when dormancy is due to hard seed coat, especially in case of legumes. Okay, so what we do here, we use H2SO4 solution for 1 to 6 minutes. Okay, also we use this in cotton scarification. Next is stratification. See, I also used to get confused. Scarification, stratification, how to remember? We get confused. So remember, like stratification, T is there. T means temperature. The other thing is opposite to that, like that you remember. Scarification, anything, C is there, chemical treatment. Here T is there, temperature. Okay, so refers to temperature treatment. Okay, so there are three types, like pre-chilling, you can give 0 to 5 degrees Celsius, example in mustard and cherry, or you can have preheating, okay, 40 to 45 degree in case of rice, or hot water treatment, okay, in case of chickpea, 80 degree for 1 to 5 minutes. The other classification is the pressure, okay, the pressure which we are applying. If you are subjugating the dormant seeds, okay, to about 2000 atmosphere pressure, about 18 degrees Celsius for 15 to 20 minutes, then this can help in breaking the dormancy. Okay, so this is usually used in seed coats are impermeable to water. There we use pressures. And another is alternating temperature. You give hot treatment, then again you give low temperature. So this will be helpful. Next is germination stimulating compounds like in gibberellic acid, kinetin. These things will also play a role, okay, in breaking dormancy. You can treat the seeds, or you can use potassium nitrate. Okay, example in case of oat. Okay, and also the urea in the gladiolus and the chichora. So this is useful in temperate zones and overcoming the extreme cold. This is very useful. In tropical zones, due to hard seed coat, improves the chance of survival. Okay, what is the significance? In temperate zones, it helps in overcoming cold. In tropical zones, seed dormancy due to hard seed coat improves the chance of survival. Okay, so next we are discussing about dormancy of cereal seeds, increase the availability of them as food. Okay, what happens is if they are not dormant, then there are concepts like vivipary. Okay, so if the plant in the plant only if it starts germinating, okay, then what will happen? You can't use it for our food, nor can you store for long. It will affect our food security. So this is very useful. That's what it is saying. So in tropical region also it's useful. Okay, improves the chance of survival because in tropical there is rain, there is temperature, many other issues, but still it will seed will be able to survive. In temperate zone, even under that cold season, it will help to overcome. Okay, so next the question is about the salt tolerance. Okay, this is means what are the how the plants tolerate this salt stress. Okay, so the morphological changes which happens is like there is poor root growth, leaf rolling, leaf tillering will be there. That will be affected. Chlorosis will be there. Leaf burning. So leaf will be having in burning patches, and the plant will be stunted compared to the bigger plant will be very small. And these all things, grain weight, these are all common things in not worry. Then next is the physiological changes which is going to happen because inhibition of photosynthesis, there will be the photosynthesis will decrease definitely. Next is the stomata will closure will happen. Okay, because there is the osmotic imbalance. Osmotic imbalance will be there. There will be high concentration of osmolytes. Okay, and there will be decreased water uptake. Understood. So these are the major factors. Biological, you can read it. High sodium transport to shoot, lower zinc and phosphorus, oxidative stress, altered metabolism. So these questions are there, and this is part of the syllabus. So in short, I'm discussing with you, but all the key points are covered, but in a very short manner. You cannot find it anywhere easily. And in drought, okay, that is also very commonly they are asked about drought. Okay, so in drought, the germination rate will be affected. The photosynthesis and chlorophyll contents are affected. There will be stomatal closure. The shoot mass rate decreases. Number of leaves decreases. Flowering, reduction in flowering. Okay, all the seed weight, seed rate, everything will decrease. So these are the issues. What are the response? Drought stress response is that there will be leaf expansion. Okay, increase in RO scavenging means ROS means reactive oxygen species. Okay, so that will increase while there is stress. Okay, and also there will be ABA production. This you know, I think, abscisic acid production, and also cells will accumulate these compounds: sugar, amino acid, alkaloid, polypols, everything. Okay, there will be osmotic adjustment which will be there. So whole process like respiration, photosynthesis, osmotic imbalance, all these things will be affected when the plant is under drought stress. Okay, so this is what it is. Next, now we will see about C3 and C4 plants. Okay, everyone have written and you know about these things. This is a very in short we'll be discussing. Okay, so what is it? Mainly the PGA is the first stable compound in C3, but in C4 we have OAA, oxaloacetic acid. Okay, and the first acceptor, okay, is RUBP, is the primary CO2 acceptor. But here in C4 plants, PP is the primary CO2 acceptor. Okay, and here we find that the diffused mesophyll and only one type of chloroplast, but here we have dimorphic chloroplast, two type of chloroplast. Okay, we there is two pigment systems present, that is PS1, PS2, but here we have in C4 plant only PS2, and we have bundle sheath cells. Okay, and in Kelvin cycle occurs in mesophyll chloroplast, but in Kelvin cycle occurs in bundle sheath chloroplast. Okay, here photo respiration is present. That's why it is less efficient. Okay, that's why it is said that if temperature increases and there is like a climate change, then the yield will decrease more in case of C3 plant. But here photorespiration is happening, so it is better. Okay, C4 plants for climate change. So like this you should link. That's why it is less efficient, more efficient. Okay, only C3 cycle is present, but here we have C3 cycle and C4 cycle. And in case of C3 plants, the optimum temperature is 10 to 25 degrees Celsius. So if it is increasing, then it will affect its yield will decrease. But here the optimum is 30 to 40 degrees Celsius. No issues. Like we have crops like sugarcane, maize, sorghum, but here we have rice, wheat, and barley. Okay, so it requires 18 ATP for one glucose molecule. Here we have 30 ATPs for one glucose molecule. This is 18 ATP. Here we have 30 ATP. Okay, so these are the very in crux, and this is, if you remember this much, I don't think you need to know much difference. Okay, so we also, I think you have read about this CAM cycle also. Okay, so what happens in case of the open stomata is in the night and the stomata closes in day. Okay, so what happens? The atmospheric CO2 comes inside, open stomata permits entry of CO2 and loss of water. Okay, then it is converted into malate, then malic acid. Okay, and this happens in the night. What happens in the morning? The malic acid, which is in the vacuole, okay, that is converted into starch. This happens in the day. So here you can see in CAM plants, tomato open at the night and closed in the night. CO2 fixation occur at the night and is called dark carbon assimilation. This is like dark carbon assimilation. Okay, like the products OAA and OAA malic acid occurs in the night. Okay, so this malic acid is produced, and during the day there is deacidification. Okay, it happens in the mesophyll cells, and then finally all the other cycles happen, then pyruvate, starch, and it happens in the chloroplast. This is the crux of the C4 cycle. Another very important question which is asked, and which was asked, is regarding aerobic respiration. Okay, aerobic respiration and anaerobic respiration. So these are all part of our notes. Okay. So here you can see aerobic respiration, oxidation of substrate in presence of oxygen, oxidation in the absence of oxygen. Okay, here we have intermolecular respiration, means between. Okay, intramolecular respiration, within. Okay, here there is complete oxidation, here there is partial oxidation, and here the output is 38 ATPs, very high energy. Okay, for higher glucose 38 ATP is produced, but here only 2 ATP is produced. Okay, here there is no, it is independent of mitochondria, it is dependent on mitochondria, because aerobic is for all the higher plants. Okay, higher organisms. This is in lower organisms. Okay, then we have the hydrogen acceptors NAD plus and FAD plus. Here NAD plus only. Okay, here both NAD plus and FAD plus. Here NAD plus only. And phosphorylation, it can be oxidative stage or and substrate level. Here only at substrate level in case of anaerobic. And in case of this is aerobic, okay, anaerobic, the respiration quotient is -1 or like more than 1, less than 1, or 0. But here it is infinity, anaerobic. And here oxidation, we have different steps. There's 6-step process, but this is just 1-step process. So this is the most crux. Like your note should be like this, that, and you not like read more. Only this much will be there, and your point should be like this. Then nobody can stop you from getting 300 plus. Okay. Next is seed viability testing methods. Okay. So though it was not directly asked, like in that test, but in previous year question also may not be directly, but indirectly you should know some 2-3 you remember, like indigo carmine test you remember, then this GADA test you remember, glutamic acid decarboxylase acidic test, which is used in maize and wheat. GADA test in maize and wheat. And then you remember radiographic method and urine sugar analysis paper test. Okay, these 4 you can remember. Just remember the name and the example, because usually they may not ask you in detail, right, what are the different methods. So just remember radiographic method, Pinus, P, and cohorts, some X-rays, and these things are used. Okay, so like that you can know. And because of these all techniques based on the color difference. Okay, for example, here you see the blue color and leaving are in unstained. Okay, so by that we can understand. So we are like there is one method of priming. Okay, so in the seed augmentation. Okay, this is also asked seed augmentation. So priming you should remember. Like only majorly you remember priming. Like we have biopriming, hydropriming, halo, osmo, solid matrix. Just remember priming. That would be sufficient. Okay, so this will be like this seed nano priming. And another next question which was asked was regarding the photosynthetic pigments. So I saw many students have written, but try to classify it like this. It looks good, na? So you have the principal components like chlorophyll A, okay. Then in bacteria chlorophyll, then we have accessory which supports the principal one. Okay, so in that we have chlorophyll B, C, D, carotenoids, phycolipins, and within carotenoids we have carotene and xanthophyll. In phycolipins we have phycoerythrin and phycocyanin. So this classification, if you write, you will get good marks. And when you write about pigments, try to draw this diagram. You will get good marks for that also, because I also saw in the paper most did not draw. So like this simple chlorophyll A, but in chlorophyll B different carotenoids and phycolipins. You can show it like this also. So different types of dormancy. Okay, like we have endogenous dormancy, wherein the seed production, the seed dormancy is because of the internal reasons. Like exogenous dormancy, it may be because of the seed coat, fruit coat, covering the endosperm prevents the seed germination, the outer portion. Okay, so reasons for seed dormancy, like it might be environmental condition, lack of water, non-availability or sufficient light, etc., temperature, etc. So we have types of seed dormancy, like physical dormancy in case of hard seed coat, lack of oxygen concentration, softening of hard seed coat. Then physiological dormancy, like non-deep, intermediate, and deep. Chemical dormancy. Okay, so we also have related dormancy. These things, just I'm telling you, you will know it. Just know the heading, that will be sufficient. Okay, we have many different types like induced dormancy, secondary dormancy, etc. This I have already discussed. Okay, scarification, stratification, hot water treatment, leaching of metabolites. Okay, temperature, you can alternate in temperature. Okay, we also can remember some of the example like this point. You can remember leaching of metabolites or inhibitors. We use coriander or sunflower. This technique is used. So here, finally, you see, I asked the question and many did not answer properly. What is the difference between plant hormones and plant growth regulators? So, what is the difference? So, plant growth hormones are chemically synthesized by plants. They are involved in plant growth and development. Okay, but plant growth regulators are chemically synthesized. They are not natural. Okay, not natural, and they are produced by humans. They are involved in growth and development, but they are not natural. Okay, here you can see the example like auxin, gibberellin, cytokinin, ABA, everything. Here we have NAA, IBA, NOA, all these things. Okay, IBA, indole butyric acid, all these things. So if you look at the synthesis, the plant hormones are synthesized as a result of plant metabolic processes. But these are because of human efforts and artificially synthesized in the lab. Okay, they are endogenous in origin, means within the plant. These are exogenous, outside we produce and we use it. In plant hormones are long lived, hence the effect is long lasting. Okay, but here the effect is short lived. The plant growth plant growth regulators. Okay, they are temporary. So this is also very, all these questions are PYQ questions, which are very important, and this will definitely help you, is what I feel. So this, just revise these things. So if I start from first, so you studied about the role of plant growth hormones, then we studied about vernalization, then we studied about breaking the dormancy. Okay, so 5 techniques I told you. One is scarification, stratification, pressure, alternating temperature, germinating compounds like gibberellic acid or kinetin, or potassium nitrate. Then I told you about the significance of sediment, whether it is required. Then I talked about salt tolerance. Okay, these are all syllabus terms, and this will give you a broad idea and very good contents. Then I told about drought stress. Okay, drought stress. Then we discussed about the C3 and C4 plants, and CAM, and aerobic and anaerobic respiration. Then the seed viability, different methods. Okay, then priming technique like the seed augmentation. We have okay, seed coating, seed pelleting, seed priming, all these things. Then photosynthetic pigments, all the different type of photosynthetic pigments. Okay. So then I finally came up with plant hormones and plant growth regulators. What are the difference? Because I didn't find many students not writing properly. So these are some of the questions which I found aspirants to be struggling. So I have made a separate slide for this. Some other questions also we have. So those things we will discuss in the next class. Some of them from the good copies of the aspirants itself. Okay, all the best. Thank you. Bye.